And now I would like to invite Claire Mérot, who is a CNRS researcher in Rennes. And before she started there, she did her postdoc in Quebec. And in general, she is interested in structural variation and their role in evolution. And what is funny that even before that, she studied her PhD and I think also undergrad here in Paris. So she basically returned to her little village where she studied for this talk. So the floor is yours, hopefully.
Hello, well, thank you. And thank you, Virginie, for organizing. It's been such a pleasure today to hear your talks. And I'd like to tell you about, so to discuss with you how genomic architecture and ecological process could interplay during evolution. And I will present you the example of chromosomal inversion in seaweed flies. Yes. Okay, so we are interested in this evolution of biodiversity at different levels, both from the genetic variation up to the diversity of species. And I guess today our point is to discuss how ecology fuels, like, enhance this diversity, and at the same time to go into the genetics, because genetic variation is truly the basis of all this biodiversity. And so in this talk, I'd like to take you to this specific level of phenotypic variation. But very often when we think about phenotypes or traits, we consider quantitative variation. But we know that there are some very discrete phenotypes, some syndromes that have been presented earlier. And I just put you here a few of quite famous examples of ecotypes or morphotypes. The rough has already been presented, but I'm showing you these beautiful butterflies, mimetic morph of Heliconus numata, but also ecotypes of Litorina or plants. And all of those examples, and many more, we see these discrete and complex types associated with a specific genetic architecture, which are chromosomal inversion. So chromosomal inversion consists in a sequence that is in a different order between two genomes, whereas there are two individuals or two species, two populations. And the peculiarity is that between the two rearranged regions, recombination will be reduced, but only in this part. So in terms of evolution or in terms of ecology, that means that there will be lots of gene flow everywhere because we are within a single species, but no gene flow in this area. So here you can easily see why this is powerful to create subdiversity within a single species. And this subdiversity is very often linked to adaptation and selected within different ecological niches, such as here joining different mimetic community in slightly different microhabitats, or in the Litorina example with different habitats favoring a crab-adaptive ecotype or a wave-adapted ecotype. But despite the high number of examples that have been arising over the last years, there are still many questions about this polymorphism. And one of them is how these polymorphisms that are discrete will persist over evolutionary time, because we could expect one ecotype to outcompete the other, for example, or the two to slowly diverge towards true species. This specific architecture will mean that these two regions of the genome will start diverging and accumulating differences, and also because recombination is lower, they're expected to accumulate deleterious effects that are more hardly purged. So we expect very specific genetic feature in this rearranged region. And of course, like to linking with selection, and one of the questions is, are there specific ecological conditions, for example, a balance between distance and ecological niches, that will particularly select this kind of architecture, or under which it would be very advantageous to have this kind of polymorphism? So I'd like to address this question in the system of Celopa frigida, and I try to take you from the field to the lab, to the genomes, and possibly back to the field, to try to understand this link between ecology, genetic architecture, and phenotypes. So the seaweed fly is this beautiful little flies that spend all its life cycle on decomposing seaweed that we call rag beds. It lays eggs, and the larvae will feed on growing microbiota. And it is present from all latitudes up to very high polar region. And it was noticed that there is lots of size polymorphism. And it's been studied a lot in the 70s and the 80s. And with cytogenetics, they could show that there are 5 polymorphic inversions, among which there is a really big one that represents 10% of the genome, and that is a complex structure. So this inversion on chromosome 1, they found the allozyme that was associated, so it was easy to score in natural population. And this showed that it has a strong impact on several traits, such as size, development time, fertility, survival, so many life history traits. So that means we have a really good idea about the link between the phenotypes and the genetics in this species. What can we learn now if we go more deeply to the genetics with the new tools that we have at our disposition? And what does it mean in terms of ecology, and how does it fit in the system? So first, when I studied this system, it was no longer so easy to do allozyme. Maybe it's never been. And we tried to develop a genetic marker, focusing on one gene, which was a marker of the inversion. We first noticed that, so when I studied the American population, they had more or less the same alleles as European population. They had the same alpha allele and beta allele, which are the 2 arrangements of the inversion. But the 2 were super divergent, about 2 or 5% in coding region. Then from one gene, we moved to the whole genome, and in this project, we sequenced along an ecological gradient about 1,000 flies. So this is in North America, and we span 1,000 kilometers, 16 population, and about 100 flies per population. We used a low coverage whole genome sequencing, which allowed to come back to each individual information, but to cover the population by about 100 individuals. So a bit like PoolSec, but with individual information. So if we try to spread genetic variation with a principal component analysis, the first thing you can notice is quite a classic pattern. The inversion strongly structure this genetic variation. It also appears when we do this kind of structure plot. If I tell the program, well, I have 2 genetic groups, which individual belongs to what? It will find the alpha-alpha, beta-beta, and the heterozygotes appears like hybrids between those 2 groups. So that means that just 10% of the genome is structuring the whole variation. Using this data, I could locate the inversion in the new genomes on chromosome 1 here, and it measures about 25 megabase. It's huge. This region is characterized by very high linkage disequilibrium, and which confirms that they are truly non-recombining haplotypic blocks. And when we look at genetic differentiation with FST between alpha-alpha and beta-beta, we see very high differentiation. You see FST up to 0.5 or 0.8. So that means that everywhere in the genomes, there is lots of gene flow. They are completely panmimetic and similar, but inside that region, the divergence is very high, like between species. And we could estimate the divergence, well, that's a rough estimate, of about 200 to 400,000 years. And in this area, there is also a strong signature of balancing selection with high Tajima-D, which is reflected by the excess of heterozygotes. So then, to get a better sense of what this was, we improved, like we did a high-quality chromosome scale assembly, both for alpha-alpha and beta-beta. And this is the chromosome 1 of alpha-alpha and beta-beta, and you can see that this region is not a simple inversion. It's not a sequence that is reversed. It's fully rearranged, with some pieces that are more divergent and some that are more similar. So I believe this corresponds to what has been described, and it could be a nested inversion with possibly different strata, a bit like a sex chromosome. But right now, we're only able to describe 2 arrangements. We annotated the genome, and this is a plot of transposable element and repeat along chromosome 1, and there is slightly more transposable element and repeat in this inversion, which could reflect a lower purging selection in this area. And there was, so we did transcriptome with EMMA, and there was more than 1,000 genes inside the inversions. Out of those 1,000 genes, about 100 are differentially expressed between karyotypes. But in the larva, there were few genes expressed, and most of them belonging to that pathway, which is involved in Drosophila in feeding behavior. So we think about this as a good candidate because of the phenotype that is observed in terms of size and development time. So overall, with this more in-depth genomic investigation, we figure out where was the chromosomal inversion and what were the genes inside. And these arrangements are really linked haplotypic blocks that are very divergent, that can be considered as supergenes with many, many genes linked together. And because of its age and high divergence, we consider that this is the characteristic of an old polymorphism that is likely maintained by balancing selection. Otherwise, we would expect that one haplotype would have fixed. And what I mean by that, when I say it's polymorphic, I mean it's not only polymorphic in one region, or you have one allele in some geographic area and not the other. This is all the population I've been sampling in Canada, and this previous work in Europe. And you can see that everywhere you will find the 3 genotypes. So it's really a cosmopolitan inversion. And nearly everywhere, we also find an excess of heterozygotes. So all of this is suggestive of balancing selection maintaining this inversion. So why this heterozygote excess? We could think about both survival and reproduction bias. First, a question that arises very often is that when you think about inversion, for example, in mammals, and think about the problem that it creates during recombination, because the 2 arrangements cannot pair, it could create unbalanced gametes. So we could think, well, the heterozygotes will have a low fitness. That should be a problem, and they should diverge. But in Diptera, well, this has been described in Drosophila, there is usually no recombination in males, and in females, you see these unbalanced gametes that are formed, they go to the polar body. So that means that the heterozygotes have as good a fitness as the other ones. And in terms of survival, they could have a much higher fitness. And this is because these 2 arrangements do not recombine, they accumulate different deleterious elements. And in the heterozygotes, because this is not the same deleterious element, they are compensated, which is called associative overdominance. And it's quite typical for this kind of big inversions. And it's exactly what we see in this species, and this has been described by Roger Butlin in the 80s. He was quantifying survival from X to adults and found a much higher survival in alpha-beta, the heterozygotes, than in alpha-alpha and beta-beta. What you can notice here is that density matters, changing the heterozygote advantage. So we could really need to consider the ecological component here. But this is not the only factors that would maintain polymorphism here. In this species, it's been noted that there is non-random and possibly disassociative mating, which is due to female choice for male, but also cryptic choice with some sperm advantage that will favor disassociative mating. So to study better that, we look into cuticular hydrocarbons with the help of Svanger, who is a chemist. And what we noticed, so here are the cuticular hydrocarbons, which are the smell of the fly cuticle. And both in Norway and Canada, you can see in yellow beta-beta and in purple alpha-alpha. And in males, it was a very different signature, meaning that they smell differently. And we expect also that females are able to perceive the difference from electroantennogram. And what we notice is that inside the inversion, there is an accumulation. Well, there are several genes which are odorant receptors or gustatory receptors. So that means that these 2 karyotypes will differ in emission and reception of chemical signal. So provided a possible mechanism on which this associative mating is based. So another reason why there is another aspect of balancing selection in this system is that I observe antagonistic pleiotropy. So what I mean by that is that fitness can be split between growth and reproduction. And what we can do is quantify the change of frequency between eggs and adults, but also between adults and eggs. So when I do that, I measured the relative survival rate, and you can notice that both the heterozygotes and beta-beta had a higher survival than alpha-alpha. And for this associative mating, when I look at the proportion in the eggs compared to what we expect under our Hardy-Weinberg assumption, we have many more alpha alleles. So that means that beta favor survival and alpha favor reproduction, which we can summarize here. So that's a classic trade-off, a life history trade-off, but here it's associated to the inversion and results in strong overdominance. So to sum up, in this inversion controls many traits, many life history traits, which result in multiple mechanisms of balancing selection: overdominance, non-associative mating, and antagonistic pleiotropy. And I think it's it's quite typical of inversion because of these many genes. So because it has so many genes, it may be not surprising to see this pleiotropic effect impacting several functions and several traits. But then we mentioned density. So what is the role of ecological factor in this context? So if we come back to that, I told you, well, it's polymorphic everywhere, but you have good eyes, and I'm sure you've noticed that it's not exactly the same proportion everywhere. So I recorded ecological characteristics of the rag bed and of the population. This is, for example, the composition of the rag bed and how deep it was. And we could associate the frequency of the inversion with the rag bed characteristic. And we see that the composition and the depth of the rag bed really explain the distribution of frequency. So this suggests that there are some kinds of rag bed that better support alpha and some rag bed that better support beta. So to test that, I went in the lab and I say, okay, I will do some experimental evolution, trying, for example, substrate like Fucasia or Laminariaceae, which are two kinds of seaweed, kelp and rag. And we measured the frequency of alpha over 5 generations, but all the replicates were showing the same. So very, very consistent, this increase in alpha frequency. If we look at the outcome, so this is a tree plot that shows the proportion of alpha-beta, alpha-alpha, beta-beta. And in black, you see the distribution in the wild. And here in purple, the distribution in our experiment. So clearly, we did something in the lab that favors alpha. But what can we learn from that? What differs between the wild and the experiment? Well, something we didn't take into account is that their habitat, the rag bed, they are ephemeral. They are removed by storms, they are removed by tides. And in the lab, I was giving them all the time they want to complete their generation. And this is important because if you look at development time, females all have the same development time. But for males, it's very variable. In short, the one that becomes big, the alpha-alpha, it takes longer. So if the rag bed is removed, they will just never reach adulthood. So this is something we need to account if we want to understand the biology of the system. And if you translate that into relative survival rate, when the habitat has a short availability, well, actually the highest survival is for beta-beta. And when it becomes longer, we see a higher survival of alpha-alpha and this overdominance that we mentioned earlier. So, and yeah, I want to add that this duration of the rag bed is very heterogeneous. It's very fluctuating over the season and from one place to the other. So to try to understand that, what are the consequences of this ecological aspect on long-term evolutionary or medium-term evolutionary outcome, we built a model with the help of Violaine. And well, this model is simply the life cycle of the flies inside the computer. So that means that instead of doing 5 generations, we can do many more and look at the outcome and the stability. And we put 2 important ecological aspects, the density, that will change the survival, and the duration of habitat availability. So what happens when we add different density to the system? So when we look at higher density, there is stronger overdominance, and this displays the equilibrium towards alpha-beta. So it does not really explain my experimental results, but maybe it explains why we have geographic variability in heterozygote excess. some population will have lots of alpha beta, and some other will be at Hardy-Weinberg equilibrium, for example. So what about the duration of the habitat availability? So if you see these are all the simulation accounting for habitat availability going from short to medium to long duration. And of course, when it's short, it favors beta-beta that develop fast, and when it's longer, it favors alpha-alpha. So here there is already this trade-off that we mentioned between survival and reproduction, and the environment, the fact that it's time limiting, even enhanced the trade-off, favoring rapid development or high survival of beta-beta, while alpha-alpha can compensate by good reproduction. And with this model, we can also think about the long-term persistence of polymorphism. So this is the frequency that we follow in the model, and if we looked after quite a lot of generation, we can ask, are we still polymorphic, like in this red area, or did we fix one allele, like beta or alpha, depending on the model parameter? And what's interesting, I think, in our question today is that, so this is how much the environment would vary in terms of duration. So in short, it's the variance. If I have one cycle with environment at 7 days and the next cycle is 20 days, this is very high variance. And the more variance we see, the more likely it is that the polymorphism is maintained. So to finish about that, like the round of the system, we found that the phenotype of different life history underlies really different syndrome, different strategy, either invest in reproduction or invest in growth, that are linked to a specific architecture with linked block that work as supergenes. And the ecology is really a habitat with temporal and spatial variation. And I believe that if we look at seasonal variation, we will see lots of fluctuation, like we just see. And this specificity, this fluctuation of the habitat, enhance this polymorphism maintenance, as we saw with the model. And someone mentioned that to me last time, and I think it kind of makes sense, that this block, this supergene, could be sometimes seen as an all-in-one solution, like providing correlated traits like we saw in the lizards. And also it could be a form of beta gene strategy. For example, the disassociative mating means that the babies would have different genotypes, so different chances, whatever the environment is. So maybe this is a process that selects for this disassociative mating. And I think I'm finished with the time. I was hoping I could tell you more fun about the next step in this system. There is we found 3 linked haplotypic blocks on chromosome 4, so more polymorphic inversion. And the cool thing about them is that they are showing a very strong climb with latitude. So we suspect, of course, that they are involved in adaptation to temperature. But that's what we are testing at the moment. And Leah Nicola is my student in Meister, and she's measuring a supercooling point, the temperature at which the fly freeze. So there is a lot of variance, but so far it seems that our inversion does not explain that. So she's measuring other traits now, and I would like to follow really the seasonal variation and decline in Europe. With that, I thank people involved, and thank you for your attention.